All right, so before I go further in this lecture, I want to clarify precision or personalized. So some time ago, like sometime means 15 years ago, people were saying personalized therapy and then it became precision therapy. So what's the actual difference between precision or personalized? Precision means precision medicines towards a subgroup of people. It's not individualized to one certain person, right? Unless the person is very rich, they can make a completely personalized therapy for them, right? If they have that much access. So personalized is used for individual patient. This is used for patient subgroups. If these are all HER2 positive, they are going to get this kind of thing. This is already widely used today. This is hardly, the whole thing is designed for one person. Biomarker is the one which drives that. Biomarker tells us which targets are overexpressed. And this is very, very relevant in oncology. This is just one whole person. We just take one person, we take all the data from that. Okay, so that is not that high throughput kind of thing. So in this lecture, basically I'm going to show you some examples, okay, where small molecules and PROTACs are used to target different enzymes. So BCR-ABL, you know, there is a chromosomal translocation. So this is, so you know, in our physiological body, the good guys become bad in cancer. So the BCR-ABL originates from ABL, and in our body it exists as c-ABL. And look at this part here. So this is switched off. So if you look at this, this is the kinase domain here. Okay, this part is another part, and these are the two domains, SH2 and SH3, just like I drew here. So it is switched, it gets open for some time, then it is switched off, then it gets open for some time, then it is switched off. So its activity is there for some time, then cells, they switch it off. But when this translocation occurs, now it becomes BCR-ABL, and what I did not tell you, BCR-ABL dimerizes. So this pink part is the BCR part. These are the SH2, SH3, and this is the kinase one. So this is always active in the BCR-ABL form. It's no switch off. So here is the thing, a good guy, if you give that person too much power, that person can become a dictator. And that's bad for the cells. So everything has to turn on when they are needed, and they need to turn off when they are not needed. But if they remain on all the time, that's when the disease occurs. Okay? So this is active all the time. But if you give Gleevec, where is Gleevec? This is Gleevec. It inhibits the kinase domain. Okay? So this is substrate binding, and this is where Gleevec binds. So this is substrate will get phosphorylated, but if you add this, substrate will not get phosphorylated. Signaling is turned off. That's why it is a $1 billion drug. But when we gave this drug, this mutation arose. First, most important mutation was this mutation. Threonine 315 was converted to isoleucine. Since then, many other mutations have been identified. But still, see, this is n is equal to 11, means 11 patients had this mutation. So this is the most common mutation, but there are many other mutations also here. Dasatinib, total 17 mutations, 17 patients had mutation. Dasatinib is the later version of Gleevec, modified. Okay, that is dasatinib. And all of these mutations, somebody asked me yesterday, what if the mutation goes out of the active site? Yeah, it does go out of the active site also. You see, this is the active site here. This is the active site. No, this is not the active site. These are the active site. But I know this is the active site because 315 is right in the middle of the active site. But the mutations are also happening in different parts of this thing. But basically, it is making that protein resistant to whatever drug we are giving. Now let's look at EGFR mutation. EGFR is a receptor tyrosine kinase. It binds EGF. This is the tyrosine domain here. So when it binds a ligand, this is normal situation. When it binds a ligand, it phosphorylates itself and turns on the signaling pathway. But when an EGFR is heavily overexpressed in lung cancer and brain cancer, okay, so now they don't even need the EGF ligand. They are just turned on all the time, okay. Gain of function, exactly. But when we gave the drug, now let's see how many mutations emerge. See the red ones, mutations emerge. That's what the problem with cancer is, that when you give a drug and it is working and the patient is happy, but then you are always praying that this person gets well before mutations arrive, and they always arrive. Sometimes they arrive as early as 3 months after treatment. Okay, so EGFR mutation, so there is a drug called osimertinib. Okay, this is a very good drug. So when you in this EGFR, there is this common mutation, T790M mutation. So when this mutation occurs, these patients get lung cancer. Okay, so look at this thing. This drug is really good, osimertinib. So this one does not bind wild type EGFR. So normal cells have wild type EGFR, it doesn't bind. So it really keeps them happy. But when there is this mutation, it binds very tightly. When there are other mutation, it also binds. So basically it is binding the bad EGFR in tumor cells. It is not touching normal cells. So this is really good news. Okay, so this is a very good, and so this overcomes this resistance here, 790. There is another mutation where exon 19 is deleted. 45% of all EGFR patients have this thing. So remember I told you about exons and introns? So one part, suppose in here you remove the tail, then this kinase will be always active, correct? So if exon 19 is removed from EGFR, it is always active. And 45% patient in lung cancer have this deletion. This drug also works for that. So we have made drugs which are specific for mutant EGFR. That's the whole point. Okay, so this drug, this one is the preferred medicine. If there is this mutation, this is the preferred medicine. So we have many drugs which are specific for cancer cells. Okay, so this is normal EGFR activation. It activates only when EGF is bound. But when there is exon 19 deletion or this mutation, they are always active. But then we have a drug which will target this and this, but not this. So we are sparing the normal cells. This is good targeted therapy, right? We are sparing normal cells, we are killing the tumor cells. Okay? So these are, I'm just talking about these mutations. These mutations are very common in lung cancer, in EGFR. Okay, why mutations predict response? Because by knowing, so now if the patient comes in, you actually do a biopsy, you do a DNA sequencing, you see exactly what's wrong with that EGFR, and then accordingly you give exactly the correct drug. So this is precision medicine because a large percentage of people have these mutations. So you have to predict, you have to actually check the mutation and then prescribe the drug, not give chemotherapy, not give chemotherapy. Okay, so this is biomarker testing. You test the exact mutations, then you exactly give the right drug, okay, depending on the mutation. And then that will also tell you what will be the prognosis for that person. Because if you have a very good drug, it's going to work very well for these patients. So this is the third generation EGF TKI. So what happens is that now cancer cells are very smart, right? So you have made drugs which can target different mutations of EGFR. So now what they will do? You are keeping EGFR shut. So now they say, okay, I don't even care about EGF. I'm going to express something else. Like you keep someone under control. So now the cells will make some other person in charge of the whole thing. So now you can keep it shut. It doesn't depend on it. This other person takes charge, and this other person is driving cancer. So when you shut down EGFR, other proteins come into play. Now they take over. So now you have to do combination therapy. You have to keep EGFR down, but you have to also kill those guys. Okay, so what happens is that now this MET kinase comes into picture. So they can be independent of EGFR, but usually when you inhibit EGFR, MET kinases also come into picture. So what happens with MET kinase alone? MET kinases, they bind this ligand called HGF, and then they get activated here. Okay, don't worry about too much details here. But in cancer, these are also activated through different kind of overexpression, fusion and things like that. And MET kinase signaling is turned on. Overexpression, or if there is any something which is keeping them in check, they delete that. So they are super active all the time. In this case, it has removed this outside thing. So it doesn't even depend on the ligand. It's always active. Dimer, always active. So many different ways of keeping it active. So if you inhibit EGFR, MET is upregulated. See, you have really made good drug for EGF receptor. So you keep it off, MET will take over. Okay. And MET is taking over, cells are going to combine. So what do you need to do it? Now you have to give a combination therapy, EGFR and MET inhibitors together. And then the tumor will regress. Okay, so this is what it's saying. You inhibit EGFR and see MET is also upregulated. Now you inhibit both of them together and you can regress the tumor. All right. Another one, ALK. ALK1 is either kinase. This will be done pretty quickly. Okay. So ALK kinase is another guy. So this is like BCR-ABL. This is also fused with some other part with other chromosome. This is super active, activates many pathways which promote cancer. All right. Feeling tired? Drink water. Just keep yourself awake. Okay, so when this is activated all the time, see here it is activated by ligand. They try to become independent of ligand so that they are always active. So this is active. Now you have to use ALK inhibitors. Okay, so you use, these are the approved inhibitors for ALK. Don't use cell phone if you are using cell phone. Okay, and then just this is inhibited. Okay. So ALK is a very important in lung cancer. So we have made several inhibitors of ALK, and then they develop resistance, and then we make inhibitors for the resistant mutants of ALK also. Okay. Last one is HER2. So you know Herceptin, which is an antibody, right? And it binds to the extracellular domain. But then this is lapatinib, it binds to the ATP binding site here and inhibits it. ATP binding site, kinase domain. So that one binds here, Herceptin, but here it is binding the active site, inhibiting it. So see, lapatinib binds here, inhibits it. Antibody binds at the outer part of the body, but lapatinib binds the kinase domain, inhibits it. Okay, so we are using many different ways to target kinases. This is BRAF mutant. BRAF is a very important protein which, yeah, go ahead.
Like if it's binding to the kinase part of the receptor, so both of the work has been saying no, like antibodies reducing signal transduction and this is doing same thing. What's the major difference?
There is no major difference. There you are adding antibody which is shutting off. Here you are giving a here it is the active site which binds ATP. So lapatinib binds to the ATP binding site and it cannot phosphorylate the protein. This is just yet another way of inhibiting it. So suppose you are giving Herceptin or something which is binding on the top and there is mutation, that is not going to work. So then you give this drug, but there is no mutation. If it develops resistance, you give this. So this is just different ways of targeting the same protein. It depends on how efficient is your antibody. It depends on how efficient is your compound. So if it develops resistance towards one, you can give the second one. Okay, so this is that famous story I was telling you about VRAF, this BRAF mutation. Remember I told you there was a lady, Indian lady with just one technician in the lab and they were working, this is that protein. And her company is the one who developed the first BRAF inhibitor. So BRAF is not bad at all, but in cancer cells there is this mutation. Valine 600 is converted to E-glutamine, and that makes this kinase super active, and that gives rise to extremely aggressive cancer. So she made the first inhibitor. Now there are many inhibitors for BRAF. So BRAF is extremely important player here. So there, see PLX, do you see PLX? PLX stands for Plexicon. That's the inhibitor made by her. Okay, just two people, she and one technician, their group made it. So there are many drugs here, okay, which inhibit VRAF. And if you inhibit VRAF, the whole signaling pathway is turned on. So this is, look at this thing. This is PET scan. Look at the cancer here and look at upon treatment how good it is. Isn't it outstanding? How metastatic cancer, I thought instead of showing you signaling pathways, I'll show you the actual thing. How bad is this cancer? Just one thing. Just see how good it is. In the brain consumes a lot of glucose, huge amount of glucose. Brain doesn't count here because our brain is anyway eating a lot of glucose. One of the ways to diagnose Alzheimer's is that Alzheimer's patients do not use that much glucose. So if glucose is missing, that means Alzheimer's is happening. Okay, but look at this cancer versus this cancer. I mean, it's just outstanding data. Inhibiting VRAF. Okay, look at this cancer and this cancer. All of them. You know, you see here all the time, this is bladder. Bladder is going to excrete it, so it's always showing. So that's not bad news. Bladder is just excretion of everything. Go ahead. Yeah, bladder is just going to excrete out all the extra things. So that's why. But just see how nice, I mean, this is just outstanding thing. Targeted therapy, how powerful it is. Okay, so this is that famous drug that women think made, right? This one mutation makes this, just one single mutation is so bad. And you give this drug and it works like a miracle. Okay. So sometimes, you know, if you are very sincere and you're doing the right thing, it can just do wonders. And you also have to have the luck to choose the right target. Okay. So this is VRAF thing. And sometimes you do that, you know, it is like you are trying to shut down one thing and the signal is going down like this. So if you also shut down the lower thing, you have a good chance of getting higher efficacy. So VRAF signals to MEG. So people have found that if you inhibit both of them, suppose you inhibit 95% of the signal here, but 5% is still leaking down. But if you inhibit 2, you are shutting off 100% signal. So if you do both, then it is even more effective. Okay, so in some cases they do that. Now VEGF and PD-1, you guys know. I know of patients who are right now undergoing PD-1 and VEGF simultaneously. So once again, I want to remind you that T cells have PD-1. So tumor cells make the ligand for PD-1 and shut it off. So T cells become quiescent. So if you give PD-L1 antibody, then T cells will bind that. Tumor cells can no longer do anything to them. Or you give PD-L1 antibody, so tumor cells cannot engage with this. So now T cells are active and they will kill it. And if you combine it with VEGF receptor inhibitor, no angiogenesis, it works like a charm. Okay, so it will completely kill tumor cells. Okay, so this combination works like a charm. Okay, so you inhibit angiogenesis, you inhibit and you turn on the immune response. And together, tumor actually, I have seen patients PET scan, it really disappears. Right before I came here to India, I watched someone's PET scan. It really works like a charm. The micrometastases in different parts of the body were disappearing with just this combination, exactly this combination. Okay, so VEGF PD-1 combination is really, really good. So combos overcome resistant, and now we also have PROTACs in it. Okay, so usually you can do monotherapy with one inhibitor, but you can always use combo drug cocktails. So that is called polypharmacology. We'll come to polypharmacology at the very end of this lecture. Not this lecture, this course. The very last topic is polypharmacology. Now let's look few examples of PROTACs. So you already know about PROTACs very well. Okay, so here this is a BCR-ABL inhibitor. Remember I told you about myristoylation site? I told you about a lot of things here, so if you recall, it's really phenomenal, okay? But BCR-ABL has a myristylation site, so this inhibitor is FDA approved. All you have to do is to bring BCR-ABL next to E3 ligase. That's all you do. So you don't care where you are anchoring BCR-ABL, as long as you bring BCR-ABL and the E3 ligase. So this is E3 ligase one blue part. This is the BCR-ABL thing. You bring them together, you degrade it. This is another compound, BCR-ABL. This is the linker. This is the E3 ligase thing. So the beauty of this is that, remember I showed you so many mutations occurring in BCR-ABL. Remember in one of the first slide. Let me go back here. Look at these many mutations occurring. Right? So if you give a drug, this mutation come, give this drug, this mutation come, give this drug, give this mutation come. But if you make the whole protein disappear, it doesn't matter how many mutations it has. The PROTAC will take care of all the mutations at once. Okay? Because you're just degrading the whole protein. However, keep in mind that you have to make sure that the mutation doesn't inhibit its binding to BCR-ABL. You have to still bind it to BCR-ABL. Okay? So these are two examples here. Then EGFR PROTACs also. So we have a lot of compounds against EGFR, against the mutants. We attach it to PROTAC, it will degrade EGFR. So these are very good. You just degrade the whole protein there. Okay, and there are many mutations there, but you can make a PROTAC, you degrade the whole protein. Okay, so that's what it is saying, that you can selectively degrade it, and usually resistance, you can overcome resistance easily than small molecule inhibitors if you degrade the, and then there are specific examples about it. Okay, so this compound, you modify it, it binds it, and then you degrade. So you can degrade many mutants of EGFR in cancer by PROTACs. Keep in mind, you don't have to remember all the details, you have to get the idea. Okay. FLT3 protein is another guy in cancer. This is also a kinase. Okay, so we have made PROTAC where we can degrade FLT3 protein. Okay, so this is FLT3 receptor. This binds the ligand and then turn on the signaling thing. So if you add inhibitors, it undergoes mutation, so the inhibitors no longer bind. All right, and then you can always use PROTAC against it. Okay, so this is just showing you FLT3's role, that it is very important in leukemia. Okay, CML, Gleevec, side effect of Gleevec is it always also inhibits FLT3. That's why it is so powerful. It not only inhibits BCR-ABL, but it also inhibits FLT3, and FLT3 is very important in leukemia. So we have made FLT3 PROTACs in 2024, which degrades FLT3. Okay, so this is just showing you the structure of FLT3 PROTAC binding. See, this is a beautiful structure. E3 ligase and FLT3 so close together, and when they are so close together, they degrade it. Okay, you guys are the real persisters. Everybody, all people disappeared except for you guys. So great job. Okay. We are coming towards the end. This is the linker, this is binding FLT3, this is binding E3 ligase. Look at that, how close they are, these two proteins, FLT3 and E3. Okay, I will stop here. Proteoforms are just, we don't have to go to proteoforms. Proteoforms are that, even proteins, same protein can exist in different forms. So they are called proteoforms, and this is extremely novel discovery. So sometimes, same protein can form different kind of structures here, proteoform A, B, C. So the beauty is that you can make a ligand exactly for a specific proteoform. So if it is able, BCR-ABL is one proteoform. If it undergoes mutation, it becomes another proteoform. So you can make specific ligand for a specific proteoform. And you can use high throughput proteomics and things like that to identify which one is overexpressed in cancer. That's what it is in the proteoform. I'll stop here. Okay. You guys can ask me questions now. We finished it early. I wanted you guys to give a break because 3 lectures at a time can be a little heavy. Go ahead. Ask me question. You had a question.
Ma'am, are we differentiating them based on the mutation or because they are proteins, these other forms are due to conformational changes?
Because the mutations, the first thing they do that they change the conformation. Okay, so for example, you have the same DNA and the same DNA can make different RNA because of splicing, that we know. So because of the splicing, we get protein 1, protein 2, protein 3, right? They are not proteoforms, they are different proteins because of splicing. But because of PTM is post-translational modifications, they can form different proteoforms here. Okay, so that's what it leads to different proteoforms. So basically what happens is that proteoform complexity matters in cancer. So for example, this protein is expressed at the cell surface, but in normal cells it is not glycosylated. In cancer it is glycosylated. So now this will bind the ligand much more tightly than this one because this has glycosylation on top of it. So imagine that this is the receptor and glycosylation is sugar attached to it. So this will change the pocket. So this will bind the ligand differently than this guy, which is not glycosylated. So that is a proteoform. Okay, if some mutation occurs, that is also a proteoform because the protein is the same, but because of the mutation, the conformation has changed. When something gets deleted, that is a proteoform because now the conformation of this protein is always active. So the point here is that we have to make a drug against a specific proteoform. So I can go through this. I can see you guys are very tired, but basically different proteoforms have different, there are different ways. For example, some proteoform will enhance proliferation, metastasis, all of those things. So if you make it against a wild type thing, wild type is not the culprit, it is the mutant or the modified one which is the culprit. So in case of SARC, this Y, this tyrosine, when it is phosphorylated, SARC is switched off. But if you change Y to phenylalanine, it is no longer phosphorylated, it is always active, right? So the cancer cells have some mutation by which they will just evade the off switch. If they evade the off switch, they are always active, and that is bad. So in a way it is good also because then you can make it specific to that proteoform. And you can do proteomics easily to identify exactly which form is there in the thing. So that is what it is telling you. The proteoformics is just an emerging field which will tell you exactly which players are involved. Okay, this is, I think, actually the last slide of this one. I was going back and forth. Should I do proteoforms? I thought I'll see at the very end. So the whole purpose of this lecture was that we have a lot of kinase inhibitors, right, by which we can inhibit the kinases. And we have used those kinase inhibitors to make the corresponding PROTACs by which we can degrade the whole protein. And both of, there are over 80 kinase inhibitors which are FDA approved. But if you compare these kinase inhibitors to which are approved in China and other countries, it's over 100 because there are many kinase inhibitors which are approved in China. Okay, I had a slide which I have not included, which included everywhere worldwide kinase inhibitors which are approved. PROTACs, they are not yet approved for kinases, but the first PROTAC is supposed to be approved in 2026 for kinase. But they are eventually going to be approved because they are already in phase 3 clinical trials, so they are working very well. And now I just showed you one way. There are many ways to make these kinase degraders. So in many cases, just inhibiting the kinase is not enough. You want to make the whole protein disappear.
In case of PARP inhibitor, like we are using PARP inhibitor, so we already have a mutation there of BRCA. So instead of inhibiting PARP, we can degrade PARP like using a PROTAC?
You can, absolutely you can. You can absolutely do that. Yeah, definitely you can do that. And the thing is that, think about it, you want to degrade this protein. It could be a kinase, it could be some other protein. Kinases give you an advantage that they have a ATP binding site. See, anything which there is a pocket, sufficiently big pocket, you can make a sufficiently nice molecule. If the pocket is not there, then how will you make a small molecule? How will you, because flat things will not give you. There you can use nanobody, because antibodies will recognize different things here, right? So you make a nanobody, and when you make a nanobody, it is a recombinant process. Nature makes nanobody. You are not making nanobody, right? You let camelid make it. So there will be trillions of molecules which are going to be formed, and then the nature will choose the best binder. So imagine making trillions of molecules. We can't do that, right? So nobody will do PhD then, right? So nature will make the nanobody, and then you attach the nanobody to a E3 ligase. Works beautifully. So so just think, enormous progress has been made. Antibodies were a problem. Now we don't even make antibodies. We make nanobodies. I love nanobodies. I really do love nanobodies, because you can, once you have the sequence for a nanobody, you don't have to go to the camelid. You can just make it in your lab using E. coli. So initially, for first time, you have to go to camelid. But once you get the nanobody, you isolate the DNA, and then you sequence it, and then you can produce in your lab. But if you're going for a new target, you of course have to go to camelid, because then there will be recombination, trillions of nanobodies will be formed, and then you will make it, right? And then just imagine how beautiful it is that you can attach two different nanobodies in one linker. So one is bringing the immune cell, one is bringing the tumor cell, and it is getting degraded. Next question. So after lunch, we have yet another really super family of proteins, like super hot family of proteins, which is called G proteins. How many of you have heard of RAS? RAS protein, RAS pathway. You have heard. You have heard. You guys have not heard of RAS pathway? Okay, RAS. Evans, you have not heard either? You have heard of RAS pathway. RAS is one guy which controls maximum number of cancers in the body. Just one single protein controls maximum number of kinases in humans. So we'll come to that, and they are very different from kinases. So we'll go and talk about those proteins. All right.